Good afternoon, everyone. My name is Jaspreet. I'm a postdoc in Dr. Eric Cohen's lab at IRCM, and today I will be presenting my work on a novel SMAC mimetic APG1387 as a latency reversing agent for HIV remission. So, what are SMACs, what are SMAC mimetics, and how do they work? SMAC is a pro-apoptotic mitochondrial protein that is released upon apoptotic stimuli into the cytosol along with cytochrome C and binds to the IAPs. SMAC antagonizes IAP-mediated caspase inhibition directly or by the proteasomal degradation of CIP1 or CIP2. SMAC mimetics are peptide-derived molecules that are based on the structure of the N-terminal tetrapeptide of native SMAC, that is AVPI. They were recently introduced as a novel therapeutic approach to treat cancer, as they help to sensitize cancer cells to apoptosis via inhibitory effect of IAPs on caspases and their capacity to activate the non-canonical NF-kappa B pathway. In context of HIV, SMAC mimetics have shown to reactivate the latently infected cells, and they may lead to the activation of apoptotic pathway to cause cell death. They may also lead to the activation of various immune clearance mechanisms. If the SMAC mimetic is not present in the system, or if the SMAC is not present in the system, the IAPs ubiquitinate various proteins and block the activation of non-canonical NF-kappa B pathway as well as apoptosis cascade. But if the SMAC or the SMAC mimetics are present, they degrade IAPs by binding to them, resulting in the accumulation of NIK, leading to the activation of non-canonical NF-kappa B pathway and releasing various caspases, leading to the caspase cascade, resulting in apoptosis. The only mechanism that remains unaffected by SMAC mimetic is the canonical NF-kappa B pathway. The aims of the study were to assess whether APG1387 can reactivate HIV in latently infected CD4 T cells and to further investigate the effect of APG on HIV reservoirs in humanized BLT mice. We started by checking the non-cytotoxic dosage of APG along with other SMAC mimetics in parental Jurkat E6.1 cells. We then checked the effect of these mimetics in the Jurkat HIV latency model such as 2D10 cells and observed that all mimetics were able to reactivate the latently infected cells at varying levels. We also observed that bivalent SMAC mimetics worked better than the monovalent SMAC mimetics. APG 1387 worked similar to AZD5582, which was recently published in Nature. Similar effects of SMAC mimetics were maintained in other GLAT models at varying levels. We then ensured the mechanism of action of APG1387 in 2D10 cells and observed the activation of non-canonical NF-kappa B pathway by looking at the conversion of P100 to P52. To further ensure that this activation was via NIK accumulation, we checked the level of latency reversal in 2D10 NIK knockout cell and observed minimal reactivation post APG and AZD treatment. Consequently, minimal effect was seen on I-kappa B, which is a hallmark protein for canonical NF-kappa B pathway. We observed significant degradation of CIP1 starting at 10 nanomolar and observed caspase activation at 0.1 micromolar. Similar mechanism of action was maintained in other GLAT models and primary CD4 T cells. We then moved to check the effect of APG1387 in primary CD4 T cells. For this, we infected the primary CD4 T cells with HIV dual reporter virus. To summarize, this virus has E2 crimson under the control of LTR, while ZS green is under the control of constitutive promoter AF1 alpha. As a result, productively infected cells will be double positive for E2 crimson as well as ZS green, while latently infected cells will only express ZS green. Further characterization of this virus can be checked on the posters by Nicholas Bellini and Isa Munoz Arias from Cohen's lab. Exploiting this tool, we studied the effect of APG in 5 donors, and an example is presented here to understand the results better. In summary, we observed no significant change in the level of productively infected cells, while a statistically significant decrease was observed in the latently infected cells post-APG treatment. We were further interested in investigating the effect of APG1387 on HIV reservoirs in humanized BLT mice. But before moving forward, it was important to check the pharmacokinetics and dynamics of APG in healthy BLT mice. For that, we divided the mice into three groups: untreated, treated with vehicle, and treated with APG1387, where we injected 20 mg per kg of the drug every third day for 4 weeks, and left the mice untreated for recovery before sacrificing it in order to see any side effects that might occur. In the interest of time, I'm not going into every result that was observed at this point, but in a nutshell, analysis of blood cells post APG 1387 treatment revealed no difference in the various immune cell population, and we also checked the non-target cell population such as B cells, and no off effect was observed. No toxicity was observed in immune cell populations of the lymphoid and the non-lymphoid tissues at sacrifice. In conclusion, APG 1387 at the dose of 20 mg per kg every third day is non-toxic over a period of 4 weeks. We further observed that the mechanism of action of APG 1387 was maintained in the tissues when analyzed ex vivo. It was interesting to see that APG 1387 was maintained in the bloodstream in between the injections, and it could also cross the blood-brain barrier successfully. Both of these results were analyzed by mass spectrometry. We then moved to investigate the effect of APG 1387 in infected ART-suppressed mice. We infected the mice with NL4.3 and checked the plasma viral load for up to 8 weeks until we were sure that viral load has reached its peak, and then injected ART on a daily basis for a period of 6 weeks to ensure viral load below the limit of detection. We then gave a single injection of APG 1387 while maintaining ART 48 hours before sacrificing the mice. We called this experiment a single dose response. We observed that plasma viral load was detected in the range of 10 raised to the power 4 RNA copies per ml of plasma in 3 of the 6 mice that received APG injections. The HIV RNA analysis of various organs revealed virus reactivation in liver and bone marrow of the APG treated group when compared to the ART alone. We also analyzed the total HIV DNA and integrated HIV DNA and observed modest decrease in the viral reservoir in almost all the organs. Consequently, we observed no difference in the proliferation and activation markers in the CD4 positive and CD8 positive T cells in various lymphoid and non-lymphoid tissues. This was an important observation, as previously it has been shown that certain SMAC mimetics can increase CD4 T cell proliferation that might act as an additional viral reservoir in the ART-suppressed SMAC-treated humanized BLT mice. The results that we observed by the single dose response of APG 1387 encouraged us to move ahead and check the multiple dose response or the cumulative dose response of APG in ART suppressed humanized BLT mice. And currently we are pursuing this study. Again, we infected the humanized BLT mice with NL4.3 virus for 8 weeks before starting the ART treatment. Once the virus is suppressed, we plan to inject 6 doses of APG over the period of 3 weeks while maintaining ART. The major analysis that we want to do in this experiment have been listed below. An interesting point that we want to check in this experiment is the extent of viral reservoir reduction at antiretroviral therapy interruption. Additionally, based on the observations made in the single dose response experiments, we are also interested in analyzing the effect of APG on Th17 cell population, as recently there were studies that suggested SMAC mimetics promote NIK-dependent inhibition of CD4 positive Th17 cell differentiation, and we think that this might be able to explain the decrease observed in HIV DNA in splenocytes without significant increase in HIV RNA. Also, in vitro analysis, APG 1387 leads to the activation of pro-apoptotic markers and activation of caspase cascade, and we want to measure the effect observed on these markers over multiple doses of APG 1387, as not much effect was observed after a single dose response. And finally, we want to check the effect of APG on myeloid cell population in various organs, as in vitro analysis of APG 1387 suggested preferential killing of latently infected THP1 cells as well as HIV dual reporter infected THP-derived macrophages, for which the results have not been shown in the interest of time. In conclusion, we observe that APG 1387 can reactivate latently infected CD4 model cell lines as well as primary CD4 T cells infected with dual reporter crimson virus. It can activate non-canonical NF-kappa B pathway via NIK accumulation and leads to the degradation of IAPs and activation of caspase 3 pathway. APG 1387 treatment is well tolerated in humanized BLT mice. It appears to induce HIV reactivation in ART-suppressed humanized BLT mice, as seen by increased viremia and augmented levels of cell-associated HIV RNA in liver, lung, and bone marrow. APG 1387 reduces the frequency of cells harboring the integrated HIV DNA in liver, spleen, bone marrow, and brain. Finally, APG 1387 is a promising approach for systemic clearance of viral reservoir that might increase opportunities for HIV eradication. I would like to end by thanking Dr. Eric Cohen and his team for the opportunity to work on this project, Dr. Eli Haddad and his team for providing the humanized BLT mice, Animal Facility at IRCM for helping by caring for the animals, Dr. Nicola Chamon and Dr. Jonathan Angel for the guidance and resources, Flow Cytometry and Proteomic Core at IRCM for their help in analysis, and all the volunteers that participated in the study. A special thank to all the funding agencies and to Ascentage Pharma for providing us with APG 1387. Thank you.